and I work at the Center of Behavioral Sciences and Mental Health in Istituto Superiore di Sanità in Rome. And these are just the recent project in which I worked. In the first one, I evaluated if absence of specific sugars of the human milk during lactation may impair attentional capabilities and cognitive function later in adulthood in mice. While now I'm working on this PRIME project that is funded by the European Union, in which where I evaluated the comorbidities between mental disturbances and cognitive function. But I will start talking about the operant system with a brief introduction about executive function. I don't know if you already know what they are or not, just a brief introduction. Essentially, they are cognitive function, high-level cognitive function that are essential to control the lower level of other function. And they entails three core components: the inhibitory control, that is essential, the capability to inhibit behaviors that no longer are useful for an objective. The working memory, so is essential to maintain a current attention for the objective. And the cognitive flexibility is essentially the ability to vary one objective compared to another. And altogether they represent executive function that are essential for the for normal life, essentially for study, for cooking, which is very important for me, that I'm Italian guy, for drive the car, and so on. And essentially the region is the prefrontal cortex, the region important for the executive function. And we also know what is this executive syndrome. Is a syndrome in which we do not have the cognitive flexibility, and this may represent one of the main symptoms is perseveratory behavior. And we can find compromised executive function in Alzheimer's disease, HIV infection, Huntington's disease, ADHD, and so on. So how executive function are studied in human, and then we will see in preclinical model. This is one of the most famous studies, the most famous test to study cognitive flexibility in human, is the Wisconsin card sorting test, and is commonly used in humans to evaluate cognitive flexibility. And essentially, take a picture, and then I will place like that because I want to show you from here.
I can send you the Thank you.
So here, it consists in a set of cards that varying for geometric shape, colors and numbers of geometric shape. And essentially the patient have to sort the cards, must be sorted by according to categories that are decided by the experimenter. So for example, number of geometric shape or color of the geometric shape. And the participants have to update and varying the rule according to the experimenter. So for example, here the participant has sorted the card for the number, so 1, 1, but the experimenter says it's wrong, it's not the number, you have to change the rule, maybe it's the color. So in the second he has to change to sort or for color or for geometric shape, and so on. And the rule is changing by the experimenter during the test. So this is another type of test, is the CANTAB, is from Cambridge, and essentially it is a tool for understand also here cognitive flexibility, and is used, for example, with the intra-extra dimensional set shift. So it's the same of the test used before, but it's without the experimenter, only with a tablet. But how cognitive flexibility is measured in preclinical models, in animal models. So this is essentially the mice version of the Wisconsin card sorting test, and it's called the attentional cell shifting task, developed in the 2000 by Birrell and Brown for rats and then adapted for mice. So essentially in this task we ask to the mice to learn a rule and then disregard this rule in favor of a new one, like the Wisconsin test. To do that, we have this apparatus with a starting compartment and two choice chamber. In every chamber we will find a bowl that is filled with digging material and odor. Underneath the digging material we will find a reward, and mice have to find this reward. Of course the mice are restricted, they passes through a restriction, caloric restriction procedure, so they want to hit the reward. So to find the reward we associate the reward with a rule, and in the first stage essentially we associated the cinnamon, the reward with cinnamon, so mice can find the sawdust in both chambers, but here for example there is sage odor, here cinnamon odor, mice have to follow cinnamon odor to find the reward. In the compound discrimination we complicated a little bit the thing, and we had the confounding factor, so another digging material is cotton here, but the reward will be still associated with cinnamon, so can be cinnamon with cotton or cinnamon with sawdust, but mice have to follow cinnamon to find the reward. In the compound discrimination reversal we changed the rule and we associated the reward with the opposite odor compared to the previous stage. In the intra-dimensional shift we change all odors and materials, but in this case we associated the reward still with an odor, in this case it's ginger. While in the extra-dimensional shift we change all odors and materials again, but the rule this time is that the reward is associated with the material and not with an odor. Okay? We measured the number of trials and the number of errors in each stage. So this is just a brief video. If work, okay, so sage with cotton, and then sawdust with cinnamon. Okay, so in this case the reward is with the cinnamon. We put the reward underneath the sawdust, and we place the bowl in one of the chamber. Of course it's all randomized, so it can be left or right, or so randomized. We place the mice in the starting compartment, and then we left the mice choose. So this is a very smart guy, he choose cinnamon. Okay, there is also a second trial in which we associated the reward with cotton. If you want to see it, just the same, but this is the second trial, so sage with sawdust, and then cotton with cinnamon, and here we place the reward in the cup with the cinnamon, also if there is cotton, but meaningless because the mice have to follow the cinnamon odor. And then I told you, he's a very smart guy and he found the rewards and very good. Okay, so why the need for OPERON? So the attentional cell shifting task is a very good task, but it has several limits. So one of these is it's very labor intensive for the experimenter, because the experimenter has to work like 5 hours per day to test 5 mice, for example. And it's, besides the labor intensive, is also very confounding for the experimenter, because I have to change every 4 minutes all the combination between the odors and the materials and the right and left chamber. So the other problem is the test is very prone to experimental errors, like inversion of the reward stimulus, or unblinded experiment that is very difficult to do with this type of study. And you know, it also needs an advanced training in behavioral neuroscience to be performed. So these smart guys developed the OPERON system. There is a very interesting system to do the attentional cell shifting task. So I will introduce a little bit on the apparatus. Then after the presentation, I will also we will do an hands-on session on the apparatus. So essentially, we have the other system here, the pellet dispenser, 2 chambers, and divided by a door. In every chamber, you can see here, you can find the magazine in which the pellet is dispensed, anomalite, 2 nose poke hole. Underneath the nose poke there is a texture floor, and above the nose poke LED lights. Texture floor are after you will see maybe better. There is a rotating wheel here then that rotate and can change the texture floor inside the apparatus. So you will have the mice will have inside the nose poke another that is pulled by this other system, and when he do the nose poke, underneath there is a tactile stimulus that change. So we managed to perform essentially the same identical task between the manual and the automated. Here you can find, you already know these, and here again we can find the cinnamon odor in the left nose poke, the sage odor in the right nose poke, the sandpaper tactile stimulus here, and the blackboard stimulus here. So 2 different confounding factors, sandpaper and blackboard, like sawdust and cotton, and 2 different odors, cinnamon and sage, like the compound discrimination of the manual. Here the right odor is cinnamon, so mice have to do a nose poke inside the hole where is cinnamon, disregarding if there is sandpaper or blackboard. So also here all the stimulus are completely randomized, and the experimenter has just to press play. So you press play and the system can change all the combination of the stimuli. Here the compound discrimination reversal is obviously the same type of materials but with a different rule. So the right odor here is sage and no more cinnamon. And also we have the extra dimensional shift in which the mice have to follow the nose poke where you find holed plastic foam compared to aluminium to have the reward. This is just another video of the OPERON system. So the mice start in the opposite chamber. Here we have infrared beam, so you see that the texture is changing. The infrared beam check that the mice is inside, so the door open it, and the mice have to do the nose poke to receive the pellet. Nose poke, but it was the wrong odor because the home lights on. So this is just an exploratory trials. It's the first trial of every stage, and in the first trial, the mice also, when he put he nose poke in the wrong hole, has to receive the reward because he has to learn that the opposite odor is right. Okay, this is just a normal trial. And you will see that mice nose poke in the right hole and receive the pellet. Okay, you see. Okay, of course, infrared beam are in each chamber, inside the nose poke hole, inside the pellet dispenser, so they monitor every movement of the mice. So what are the pros and the cons of both this type of test? For the attentional cell shifting task, the pros has an high ethological validity because mice love to dig, love to smell, and it needs also less animal to be performed, and it's highly represented in scientific. But the cons is, as we seen before, it's very labor intensive, it's prone to experimental errors, and it needs an advanced training. While with the OPERON system, we have a reduced percentage of experimental bias and errors, limited manpower because you only have to press play and stop, and also lower level of training in behavioral neuroscience. And the cons is not a cons of the system, but of every operant chamber. So there is a lower ethological validity and also higher number of experimental dropout compared to the manual. But it's not cons of the operant system; it's a cons of every type of this test. So I will conclude the presentation with a little type of experiment that we have performed in Rome, so a validation of this system. To validate, we use two different strains of mice, CD1 and C57, because we know that in literature there are strain differences in attentional capabilities between these two types of strains. And we have these 4 experimental groups, so CD1 performing manual attentional and CD1 performing the OPERON, and then C57 performing the manual and C57 performing the OPERON. So what we see essentially is that OPERON system bestows results analogous to the attentional set shifting, because we can see that there are differences between within strains. For example, here we found that C57 completed the CDR stage with more difficulties compared to the CD stage, because CDR is more difficult for mice. We see that in the manual they perform more trials and more errors in the CDR compared to the CD, and these results are exactly the same in the OPERON system. So the C57 mice committed more trial and errors compared to the CD. Then we also found the same result in the intradimensional shift. So the intradimensional shift is simpler compared to the compound discrimination reversal in both type of the test. And then we also find